So it's our great pleasure to invite Professor Sugiyama as a lecturer, plenary lecturer. He received a PhD in Kyoto University, and now he's a professor in Kyoto University, and also he's a president of Institute of Integrated Cell Material Science, ICMS. Also, last year he received Japanese Chemical Society Award from the Chemical Society of Japan. He's very famous in construction of innovative biomaterials.
Today I would like to talk about the chemical biology of nucleic acid and mainly focus on the DNA origami and artificial genetic switch. So let me start with DNA origami. The origami, as you know, the famous hobby and craft, popular craft. Thank you. Craft, especially in Asian country. So we can make many kind of shape using just a sheet of paper, like crane or frog. Now the people gradually realized that DNA can be used for that purpose too. So this is a first DNA origami paper published in Nature in 2006 by Paul Rosamond. And this tall person is Paul Rosamond. Sorry. And he only used specific Watson-Crick base pair and the canonical structure of B-form DNA. He demonstrated that he can create many shapes, including smiley face. Since this method, DNA origami method, is very reliable and robust, therefore various two-dimensional as well as three-dimensional structure have been prepared using this method. Let me show how to make DNA smiley face. First, we need to make the pattern of smiley face using M13 DNA sequence. If we set the starting point, then all the place we know the sequence. Then we place the complementary strand, colored in green, and they are different. So let's take a look at this section. We have M13 DNA here. So in order to stabilize this, we place the staple like this by forming Watson-Crick base pair. So 8 base pair stabilize this duplex, and another 8 base pair stabilize this region. Overall 30 tumor staple fix this part. Of course, other places have different sequence, overall 226 different staples to keep this structure. And we mix with M13 DNA with this staple and heat it up at 85 degrees C and cool down to room temperature. Then we can get the smiley face. So this animation was made by Harvard Group, where you can see the short staples find the complementary strand, a complementary region of long DNA fragment, and gradually forms the rectangle structure. I hope you can get the image of how the origami forms in the solution. For visualization, we use high-speed atomic force microscope because the molecule is very small. This is 100 nanometer size. So this is a Rosamond designed DNA rectangle on microsurface in solution. So during the measurement, so this part is actually the extra loop region down there is moving or rotating. That clearly indicates the power of high-speed AFM. If we want to make a 3D structure, we often prepare the extended form first. So this is a case of DNA box. You can see the extended structure here. And by adding the connecting strand, which connect this face and this face, and this face and this face, so on, then we can get the particle. And the size is reasonable based on light dynamic scattering analysis. And this is the image of AFM. Please look at this one. This one we think is a one-phase open structure. And gradually open another phase. And it takes about 10 seconds to open the complete structure. One could imagine if we put the RNA or toxin or protein inside the box, probably this method can be used for the new type of drug delivery system. So since we are organic chemists, we'd like to make something useful using origami technique. And we prepare the DNA frame shown here, on which have hole in the center with 2 DNA bridges. These DNA bridges are for the substrate for DNA modifying enzyme like recombinase and repair enzyme and so on. And also DNA, these DNA bridges, they can be the substrate for DNA structure change. And I'd like to show some of the example. The first example is the Meso Transferase. This enzyme put methyl group at adenine of GAA-TTC sequence. So both bridge has that methylation sequence in the center. And the top strand has 64 base pair that fit between A and B. But bottom strand has 74 that has 10 extra rings to make a kind of relaxed duplex. And we looked at the reactivity. So you can see the enzyme bind to one of the bridge in solution of microsurface and sliding along the helix. It seems the enzyme is looking for the recognition sequence for methylation. And this is actually the direct observation of single molecular dynamics of enzyme in 40 by 40 nanometer space. And the important question is how the methylation reaction occurred in tensed or relaxed duplex. And we found relaxed duplex is better substrate for methylation, about 3 times more reactive than the tensed duplex. Next example is recombination. Recombination, as you know, the very important biological process to create new genome from the genome from father and mother. It was proposed that recombinase bind to the substrate to set of duplex strand as a tetramer and resolve it. White spot you can see in the center is a recombinase, and the tetramer-like shape is a little bit clearer, and all of a sudden they dissociate into monomers, leaving the double loop structure as a recombination product. So this is a direct observation of the recombination process using DNA frame. Here we control the orientation of the DNA using DNA frame. The previous case, we control the tension of DNA. And 2 years ago, my colleague in plant biology discovered the MOC1 protein. That enzyme is essential for segregation of chloroplast. And he hypothesized that this enzyme resolves the holiday junction type structure and resolve it. And using our platform, we could visualize protein binding here and resolvation by this MOC1 protein. Our next example is the Cas9 cleavage. The Cas9, as you know, is a very powerful tool in gene editing now. And we can visualize the sequence selective cleavage using our platform. And the white spot is Cas9 with guide RNA. And we introduced a rotational constraint of the target strand, which forms a duplex with guide RNA, and also non-target strand. Then we found that rotational constraint on the non-target strand reduced the cleavage efficiency of the Cas9, clearly indicates the rotation of non-target strand is very important here. And several years ago, our group and 2 other groups in United States elucidate the topology of human telomere sequence as shown here, which consists of the triple helix and single strand. And we propose that such G quadruplex can stabilize a relatively long single strand region based on the gel electrophoresis and also melting temperature experiment. However, those are very indirect. Therefore, we decided to use platform and introduce the single repeat on top strand in DNA frame and the bottom strand, 3 repeat on bottom strand, and ask whether it forms the X-shaped structure in the presence of potassium ion. So this is an image. In the absence of potassium ion, you can see the parallel DNA. And in the presence of potassium ion, you can see the formation of X-shaped structure. So this is a close-up view of one of the DNA frame. And during the measurement, we introduced the potassium ion. You can see the parallel DNA bridge suddenly forms the X-shaped structure and clearly indicates the formation of G quadruplex at single molecular level. And recently, our group also used DNA origami for a different purpose too. So this is a collaborative work with Professor Hanbin Mao at Kent State University. He is very good at the optical tweezers. And we place the G quadruplex between 2 beads. And measure the force and extension counter length by force extension curve shown here. And now we put this G quadruplex inside the DNA nanocage made of DNA and look at whether that nanocage stabilizes the quadruplex or not. And this is a result. Without nanocage, the counter length is about 8.3 nanometer, and unfolding force is 20.7 piconewton. However, inside the nanocage, the counter length stays constant. However, unfolding force is almost double, clearly indicate the nanocage stabilizes the G quadruplex. And we also place the I motif, so complementary sequence of the G-rich strand can form the I motif. That structure is also the quadruplex structure. And without nanocage and within nanocage, the counter length again is almost the same. However, unfolding force are significantly stronger in the nanocage. And we changed the size of nanocage and plotted the effect here and here. So, and we conclude that the overall loss of water drives the folding of tetraplex in nanocage with reduced water activity. So this is a kind of summary of the origami project, and I hope I could convince you DNA origami is powerful technique to visualize the single molecular event. So let me move to the next topic of artificial genetic switch. So this is a cartoon of the cell. As you know, the gene expression is carefully and precisely controlled through DNA-protein interaction, DNA protein and protein-protein interaction. So it would be nice if we can design the molecule which can control the DNA gene expression on demand, and therefore our group is focusing on the creation of artificial genetic switch based on the chemical synthesis and molecular biology. The molecule which we are interested in as a building block of artificial genetic switch is the fluoroimidazole polyamide developed by mainly Peter Dervan at Caltech. The molecule consists of N-methyl imidazole and N-methyl fluoro, the kind of peptide molecule. By introducing aminobutyric acid, the molecule fold into hairpin and tightly bind to the minor group of helix in sequence-specific fashion. The sequence specificity is based on very simple recognition rule, that is a pair of imidazole fluoro recognize GC base pair, and the fluoro pair recognize AT or TA. Fluoroimidazole pair recognize CG. So one can easily synthesize a molecule which can bind the specific sequence. Also, aminobutyric site and beta-alanine part recognize the AT base pair. This small molecule recognize 6 base pair rings. Now, one of the advantages of the molecule is good nuclear permeability. So this is the image of 2 hours treatment of 1 nanomolar concentration of FITC labeled polyamide shown here. As you can see, all the nucleus of human vascular smooth muscle cell are stained under the condition. And I'd like to discuss off-switch by simple binding of polyamide first. So again, this is a cartoon of the gene expression. As you know, the transcription factors bind to the promoter region and recruit the RNA polymerase to start the transcription. Since the polyamide can be designed to any sequence, so let's design here, then polyamide may inhibit the transcription factor binding. But as I mentioned, the polyamide binds to the minor groove of the helix. Why the transcription factor cannot bind? Because they use a major groove surface. So this is a crystallographic analysis of the minor groove. As you can see, after polyamide binding, minor groove gets wider. As a result, major groove gets narrower, and therefore, binding of polyamide to the minor groove affects the inhibit the transcription factor binding. And I'd like to show one of the example of off-switch. This is SOX2 inhibiting polyamide. As you know, the induced pluripotent stem cell developed by Professor Yamanaka can differentiate into many kind of cells. But, they usually are very unstable, easy to differentiate to mesoderm and ectoderm and so on. For example, SOX2 actually prevents the differentiation into mesoderm. Therefore, we can assume that if we inhibit the SOX2, iPS cell would be differentiate into mesoderm. If we added the SOX2, then we can see the shifted band here. Upon addition of designed polyamide here, the amount of DNA SOX2 band decreased with the formation of PIP DNA. Clearly indicates the polyamide can replace the SOX2 about 5-fold excess. And we treated the iPS cell with this polyamide. After 5 days, expression of mesoderm specific gene like T. brachyuri, and GATA4 are highly expressed. And SOX2 expression going down. And we can confirm such effect using immunostaining. So polyamide inhibit the SOX2 here and upregulation of brachyury. That clearly indicates the iPS cell goes to the mesoderm. And usually mesoderm can be converted to beating cardiomyocyte using Wnt signaling inhibitor like IWP2 shown here. So we treated the iPS cell with this polyamide and treat with IWP2, then we can get the beating cardiomyocytes shown here. And the number of the colony of beating cell is quite good, and also cardiomyocyte markers are expressed that confirm this is a cardiomyocyte. And the second step, in this case, we use IWP2. However, this step can also can be replaced with the polyamide shown here by inhibiting the TCF binding. So these results suggest that other differentiation, like the differentiate into neuron or neural cell, can be also possible with using the combinatorial use of polyamides. And usually polyamide goes to nucleus. However, if we conjugate the mitochondria penetrating peptide shown here, then molecule goes to mitochondria. co-staining clearly confirmed the polyamide goes to the mitochondria and suppress the mitochondrial gene of ND6. So polyamide can be used to control the mitochondrial gene expression too. So let me move to alkylation, off-switch by alkylation. So in the case of alkylating agent, we can prevent the transcription factor binding, and also we can stop the transcription during the transcription process by polymerase. Therefore, our group synthesized the various type of sequence selective alkylating agent shown here, and by introducing various motifs, now we can synthesize the sequence specific alkylating polyamide with 12 base pair recognition. And I'd like to show some of the example. First one is inhibition of KRAS. KRAS is one of the most frequently activated oncogene. A mutation causes the uncontrollable proliferation. Actual major mutation is codon 12 of GGT and to GTT. So this is a major mutation. Therefore, we synthesize the polyamide which selectively target the mutated GTT sequence. And sequencing gel clearly indicate the molecule selectively alkylate one match site and effectively reduce the expression of KRAS. And we synthesize this compound in large quantity and inject the pneumomas which carry human colon cancer. As you can see, after 5 weeks after injection of this compound, we can get the smooth surface and clearly indicate the molecule reduce the growth of cancer without any loss of weight. Next example is inhibition of ranks 1 to 3 by polyamide. Dr. Kamikubo at Medical School of Kyoto University proposed that ranks 1 to 3 are responsible for malignancy of acute lymphoma. Ranks form the heterodimer to drive the BCR-ABL and MLL fusion protein. Therefore, we synthesize this compound to prevent the binding of ranks 1 to 3. The molecule selectively alkylate the target adenines, shown here by gel electrophoresis, and identify the adduct using the oligonucleotide experiment. The molecule showed the relatively strong cytotoxicity against various human cancer cells, including AML, ALL, colon cancer, breast cancer, and so on. However, some other groups didn't show the strong efficacy. The Chromobacillus M prime shows strong cytotoxicity against a cancer cell with wild type p53. That may be probably p53 may be responsible for strong cytotoxicity here. And we propose that ranks 1 regulate TRIM24 and BCL11A, those degrade p53 that explain this phenomena. We similarly set up the animal model of acute myeloid leukemia, shown here. The animal mouse died with within 20 days. However, injecting this compound from tail vein extends the life period about 20 days. The conventional RRC only has a mild effect, clearly indicate this is quite effective. And also, it is more important to say that chlorambucil S, which have the same alkylating agent, but a different sequence specificity, did not show any positive effect, clearly indicate the importance of the recognition sequence of polyamide. And such type of extension was also observed the acute lymphoblastic leukemia extended about 20 days. These results suggest the significant extension of survival period of AML and ALL mouse model. Also, the compound effective against lung cancer and stomach cancer. Our molecule binds to the minor groove in a sequence-specific fashion and make covalent bond with DNA and shut down the function of DNA. And this is a typical off switch. Let's move to on switch. So in order to switch on a gene expression, we need to consider epigenetics because transcription factor can bind to loose chromatin structure. So easiest way is probably the inhibition of HDAC. And this is the concept of our on switch. We conjugated the HDAC inhibitor SAHA at the end of the polyamide and try to deliver the HDAC inhibitor locally and recruit histone acetyl transferase. And they put the acetyl group and neutralize the electrostatic interaction to get the gene activation. So this is a proposal. And we synthesized the various small library and analyzed the expression and found that each SAHA conjugate activates a different set of genes. So this is a typical example. K SAHA conjugate activate germ cell genes, and I activate pluripotency gene, and K X activate retinal gene, and so on. And we made a video, and this part is HDAC inhibitor. So polyamide carry this warhead to the DNA in the nucleus and the nucleosome context, and the molecule bind to the minor groove in the sequence-specific fashion. And they inhibit the HDAC colored in green, and then histone acetyl transferase can put the acetyl group and neutralize the electrostatic interaction, and then transcription factors can bind and finally recruit the RNA polymerase to start the transcription. So this is hypothesis. So in this movie, in this scenario, we inhibit the HDAC and indirectly activate the hat to make euchromatin from heterochromatin. The question may arise that can we directly activate the histone acetyl transferase to get the similar effect? And if we look at the literature, there is one compound, CTB, activate the histone acetyl transferase. To test this hypothesis, we synthesized the polyamide conjugate shown here and compare the efficacy with SAHA derivative. And this is microarray data. The monomeric CTB and the SAHA activate the different set of genes. However, once it conjugate to the polyamide I, they activate the pluripotency genes. Sorry. These results indicate the importance of polyamide and also the activation of HAT can cause the similar activation as HDAC inhibition shown here. And recently, our group synthesized another type of on switch that is a bromodomain inhibitor PIP conjugate. So this is the structure of the P300. In addition to catalytic domain, there are many domains, including bromodomain. And we can draw the enzyme like this. A bromodomain, actually, they recognize the acetylated lysine residue of histones and put another acetyl group in order to expand the euchromatin region. So we synthesized a bromodomain inhibitor polyamide conjugate, shown here. So this part tightly binds to bromodomain. And we expect the polyamide recruited P300 to put acetyl group near the binding site. And we use a reconstituted nucleosome system to look at the acetylation level. And the BIPIP1, shown here, induced the acetylation only on nuc1. Actually, nuc2 has a binding site for PIP2. And we treated the hex cell with PIP1 and PIP2 and analyzed the expression, and we found 1 and 2 activate individual 200 genes. And this is a volcano map, and we selected the typical activated gene of NTS by 1 and IER5L by 2, and confirm the activation by using QRT PCR. And we confirm the activation. And if we look at the binding site, PIP1 has many binding sites for NTS, but no binding site for 2. And whereas IER5L has 3 match sites for PIP3, that clearly indicate the binding can explain the selective gene activation. And let me move to the final, cooperative binding. As I showed, the gene expression system is very, very complicated. And they actually use cooperative energy. Since the recognition sequence of the transcription factor itself is not so long, like 6 base pair or 8 base pair, sometimes 4. But they interact each other to perform the precise binding. So important question is, can we install the cooperative interaction into polyamide? So we just tested that hypothesis. We synthesized cyclodextrin and adamantine conjugate, and whether they can bind to the target sequence cooperatively or not. We estimated the cooperativity using the melting temperature and also gel shift shown here. This is a complex formation of the monomeric part. If we use both of them, you can see at much lower concentration, we can see the complex formation, clearly indicate that both polyamide cooperatively bind to the target strand. And we calculated the cooperative energy as minus 2.3 kcal, which is about the same as the natural transcription system. And we tested the efficacy in the living cell using the reporter system, which have estrogen receptor promoter region. And this is a molecule, these are the molecule, and each monomer reduce the expression of firefly. However, if we use together, then very strong inhibition was observed that clearly indicate the cooperative assembly can be utilized in the cell. So this is a summary, and first origami part was led by Dr. Endo, and the second part was led by Dr. Bando and Dr. Ganesh Pandian. Also, Soyang Park in the audience contributed in the synthesis of fluorescent labeled DNA. Without their dedication and enthusiasm for science and hard work, the presentation today would not have been possible. Thank you very much for your kind attention.
Thank you, Professor Sugiyama. So you beautifully shows how you can control DNA protein interaction using small molecule, PIP molecule. So this is a very great work, but I would like to ask another opportunities, which is RNA and protein or RNA biomolecule interaction. So the RNA these days is a very hot topic, especially the microRNA or non-coding RNA interaction with the protein. So basically, from logical guesses, we can expand your P molecule to target the RNA. Then it's your work over the period advance and community works would make a big impact. So can you make a comments? And also I have the minor questions. So eventually, if you want to translate this technology into in vivo, so in vivo drug delivery in vivo delivery would be essential the problem, right? So I'm just curious as to how you can specifically target the specific location of inside of human body, right? So the long-term circulation is randomly your P molecules can deliver to any non-specific region. So how you can design your P molecule only target the cancer location or the inflammation or the injury local site.
Can we design the RNA binding polyamide? We investigate the binding of polyamide to the RNA DNA duplex or RNA duplex. The polyamide usually doesn't bind well. So binding constant is maybe 100-fold less than targeting DNA. And we also tried the in vitro selection of the polyamide, but we couldn't get positive data yet. But the RNA has many structures, so probably you can find the good inhibitor in the library. And the second question of the drug delivery system, our molecule, the polyamide, has a tendency to accumulate the cancer. We don't know the reason, but FITC level of polyamide clearly indicate after injection, it quickly disappeared from the blood, but it accumulates the cancer cells. So I think there is some affinity to the cancer cells. And but recent drug delivery system are very much improved, and we would like to try your nano script or nanoparticle or liposome or even origami structure.
Okay, thank you.
Thank you.